All right, everyone. In this video, we're going to do the first part of our discussion of glycogen degradation. The first video will talk about the enzymes associated with this, and then in the second part of the series, we will look at the regulation. Here is our metabolic map, and we have been talking about catabolism and really breaking down these molecules to give us energy, but we are going to now jump back up here and look at this energy storage piece, right? So here's our glycogen, and if we are bringing this back over to jump into our glycolysis, jump into our gluconeogenesis, we're going to come down here. And then if we need to, we can get back up to our glucose. All right, so we can break this down into its glucose monomers. So just briefly about glycogen, it is a highly branched homopolymer of glucose. So homopolymer, all the monomers are the same. So all the monomers are glucose. Okay, this is also present in things like bacteria. Plants store their glucose as starch instead, so a little bit different. The largest stores of glycogen are in the liver and the muscle, and glycogen functions differ based on where in the body they're found. Okay, the liver is going to break down the glycogen and release glucose to the bloodstream to provide energy for the brain and red blood cells. Okay, versus muscle glycogen stores are generally going to be mobilized to provide energy for muscle contraction, right? These are energy stores for when you need to run, you need to move. If we look at how it is stored, this glycogen is in our body, it's in these granules. So this picture on the left is this granule. Those granules tend to be about 10 to 40 nanometers in diameter. They tend to have somewhere around 55,000 glucose per granule. Okay, so these are pretty big granules. Over here on the right, we're looking at a cross section. And this is really meant to show you kind of these branching, right? So it is a highly branched polymer. Right in the middle here, and we'll talk about this more next week, but right in the middle we have this G protein, okay, glycogenin. So it's a G protein, and when we talk about glycogen synthesis, we'll look at this. This is kind of a primer to get the glycogen started. All right, so let's look at it now on more of a molecular level. So starting up here at the top, here is our glycogen. Lots and lots and lots of glucose monomer units. And we can see the branching, right? So we've got a straight chain polymer. It's not necessarily straight. It's going to have kinks and turns, right? But overall, these are all in a line. And then this glucose monomer has two connections, right? There's our branch. When we look, these connections are alpha 1,4 linkages. So here was our first, oh, and you can see we've even got them numbered for you over here. Here was carbon 1. It's that anomeric carbon. Remember that term? Anomeric carbon is the first carbon. And then it is linked to carbon 4 on the other. So we have these alpha 1,4 linkages. If we look at our branches up here, now they're alpha 1,6. So the alpha, the anomeric carbon from that branched one, is linking to that 6 carbon in our straight chain. Over here, you'll notice that we have labeled the non-reducing ends. you can identify the non-reducing ends because it is the end that does not have the anomeric carbon free. So over here on the other side, right, this is that anomeric carbon. But our non-reducing ends, that end there is not the anomeric carbon, okay? And it's important because our enzymes are going to break down the glycogen working from the non-reducing ends. So when you think about, in the past we talked about proteases that snip proteins in various places, like chymotrypsin, trypsin. So the glycogen phosphorylase, which we're about to look at, is the enzyme that's going to break down these alpha 1,4 linkages. So glycogen phosphorylase can break them, but it only starts at the non-reducing ends and clips off one at a time. All right, if we look at the bottom, we can look at that. So what we have down here is a very small piece of one of our glycogens. It's not showing any of the branches. And our glycogen phosphorylase, here, I'll write that even though it will be on the future slide. So our glycogen phosphorylase, which is oftentimes just called phosphorylase. Okay, that glycogen phosphorylase is going to add a phosphate group across that first alpha 1,4 linkage. So we see that over here. We've added the phosphate and we've broken that bond. So these are similar to hydrolases. So oftentimes the enzymes that break apart these big biopolymers are hydrolases. They add water. So instead of this phosphate group right here, we'd have an OH group for a hydrolase. But glycogen phosphorylase is going to add that phosphate group. All right, let's take a look at kind of a little bit of the intricacies of this. We need several enzymes. So why do we need several enzymes? Seems like it's all glycogen, or it's all glucose monomers. The glycogen phosphorylase should work. And the key is that the phosphorylase will work, but only on straight chain pieces. So if we look at our glycogen up here, so each of these hexagons is a glucose monomer. Okay, the color coding is just for us to be able to view how the phosphorylase works. Our phosphorylase will start at the non-reducing end, which is over here on the left. Okay, I'm just telling you that. And our phosphorylase could work on this chain and on this chain simultaneously, right? Well, different phosphorylase active sites could work on these two chains. And they will keep going until they reach 4 residues from the branch, right? So if I look at my numbering here, 1, 2, 3, 4. So it's going to stop at that one we've highlighted in blue. 1, 2, 3, 4. It's going to stop when it gets to that one we've shown in yellow here. So 4, not including the one that it's branching from. Okay, it can act up until that point. Each time it's snipping one of these off, it's giving us a glucose 1 phosphate, which is great, but that's not what we need. We need that glucose 6 phosphate so it can go into glycose or glycolysis or into what we're going to call the pentose phosphate pathway to get other carbs. We'll take a look at that next week. Or back to glucose. So anyway, we need to get this to glucose 1,6-phosphate. We have an enzyme that will do that for us. I'm sorry, not glucose 1,6-phosphate, glucose 6-phosphate. Okay, that enzyme is phosphoglucomutase. Okay, and that can happen in any organ. So we'll snip off these individual glucose 1 phosphates, and we will, so you saw that right up here. And then we have a different enzyme that will convert them to the glucose 6 phosphate that I misspelled there, that you can see in our various pathways. But now we have this molecule, and we would like to continue being able to break this down and getting our glucose back. So we need 2 other enzymes to rearrange, or what we often call remodel, this glycogen. Okay, the first one is a transferase, and what it does is it snips off this piece and then reattaches it down here to this other end. So now we've got this longer straight chain. Our phosphorylase could come back in here, right? And start working on it again. And it could work on it again until it got to 1, 2, 3, 4, till it got back to that yellow. So that will take care of part of our problem. But then we're going to be stuck again because we still have this branch. And so what we do there is we're going to snip off this glucose. Okay, our enzyme is alpha-1,6-glucosidase, and it's going to, it's a hydrolase, it's going to add water to separate that glucose. And then because free glucose isn't really desirable for metabolic processes in the cell because it can leave the cell, our hexokinase is going to come in and convert that into the glucose 6 phosphate. So we have, essentially, when we look at it, 3 enzymes needed to break down our glycogen. And then we need to get those pieces over to our glucose 6 phosphate so they can drop into our other metabolic pathways. I want to take a minute and just look at this enzyme. So let's orient ourselves here. This is just our big enzyme protein. Here's the important little piece in our active site, which is the serine residue. Okay, and when we look at it, that serine residue in this enzyme has a phosphate group attached. And what happens is our glucose 1 phosphate drops into this active site, and that serine residue transfers its phosphate over to the molecule. But then this molecule still in the active site gives back a phosphate, but it gives back the other one. Okay, and so in that way we have now transferred. So this is an isomerase. It's going to change where that phosphate group is. It rearranges it. Okay, so we looked at, so the liver contains glucose 6 phosphatase, a hydrolytic enzyme that's absent from the muscle. So when we come back up here, right, we see that as a product of this, that is not what I want. There's what I want. All right. When I look at this, I have made glucose 6 phosphate. I have made glucose 6 phosphate. So when we come back down here, just as kind of an aside, this glucose 6 phosphatase, this enzyme, is going to generate that free glucose. It's going to snip off that phosphate group in the liver. And then that free glucose can be released into the blood so that it can be used by your brain, your red blood cells. And this is just kind of a point, looking at how things have evolved. Glucose 6 phosphatase is absent in most other tissues. It's really only there in liver, and that's because our liver cells are the cells that are tasked with keeping our blood glucose levels constant, right? So this phosphatase enzyme that removes the phosphate group, and now that we've made that glucose 6-phosphate, if we need just glucose, it can snip off that phosphate. All right, in our next video, we will look at the regulation of this whole cycle.